Welcome back to the deep dive. Today we are diving into mirror life. And I have to be honest with you. When I first saw this stack of research come across the desk, the reports from Science, the transcripts from the Paris conference, my brain didn't initially go to biology. It went straight to science fiction. I'm thinking Star Trek, the mirror universe.
Naturally. It's the first place everyone goes. It's iconic.
You know the one, right? Where Spock has the goatee, the Federation is this terrifying empire, and everyone is the evil version of themselves.
The evil twin trope, a classic.
Exactly. Or, even Alice in Wonderland stepping through the looking glass into a world where logic is inverted and everything is just backwards. It feels like pure fantasy.
It feels that way because it is so counterintuitive to our daily experience. And honestly, those stories are great fun. But what we are talking about today isn't about parallel quantum dimensions or evil twins with goatees.
Oh, no evil Spock.
No evil Spock.
Yeah.
We aren't talking about magic. We are talking about geometry.
Geometry, that seems like a much drier subject than evil twins.
You might think so, but it's actually the geometry of life itself. Because while evil Spock is definitely fiction, the concept of a mirror world of biology, a world where the chemistry of life is physically flipped in reverse, is scientifically possible.
Okay, scientifically possible is a big phrase.
It is. In fact, looking at the milestones we've hit in just the last 12 months, it's not just possible. We are standing right on the edge of being able to create it.
That is a massive statement. So just to set the stage for everyone listening, it is Tuesday, January 27, 2026. And the reason we're doing this deep dive now is because over the last year, this topic has migrated from, obscure academic journals to the front page. We had that massive report in Science back in December 2024, and then the emergency conference in Paris last June.
Exactly. This has moved from cool sci-fi idea to serious scientific reality very, very quickly. And the big question we're grappling with today is simple, but, well, it's mind-bending. What if we built life, bacteria, cells, maybe eventually plants, that was chemically identical to life on Earth, but structurally flipped in reverse?
Like a mirror image that actually breathes and grows?
Precisely. A biological reflection that is physically real.
Okay, let's unpack this. Because to understand how we can flip life, we first have to understand that life has a direction, or a shape to begin with. The papers you sent over refer to this as the handshake problem.
Right, and this is the core concept. If you get this, you get everything else we're going to discuss regarding the risks and the benefits. I want everyone listening to try something right now. Unless you're driving, please keep your hands on the wheel. But if you can, hold your hands out in front of you, palms facing up.
Okay, I'm doing it. Palms up, staring at my hands.
Now, look at them. They look identical, right?
Yeah.
4 fingers, 1 thumb, the lines on your palms. They are perfect mirror images of each other.
Sure, they match. If I put them side by side, they look like twins.
Now, try to put one on top of the other. Put your left hand on top of your right hand, both palms facing down. Do they line up?
No, not at all. My thumbs are sticking out on opposite sides. My pinky is hovering over my index finger. They don't stack. It feels weird, wrong.
Exactly. In geometry, we call this being non-superimposable. You cannot make your left hand fit into the space of your right hand unless you flip it over. This property, where an object has a mirror image that can't be superimposed on the original, is called chirality.
Chirality.
It comes from the Greek word care, meaning hand. And the crucial thing to understand is that it's not just hands, it's molecules. The fundamental building blocks of life are chiral. They have a shape, and that shape has a direction.
So molecules can be left-handed or right-handed, like my hands.
They can. And here's the best way to visualize why that matters. Imagine meeting someone new. You reach out your right hand to shake their hand, but they reach out their left hand.
Oh, that is the worst. It's that awkward fumble where you grab their fingers and you don't know what to do.
It's incredibly awkward. You're grabbing their wrist, your thumbs are clashing, it just doesn't fit. That is the handshake problem. In biology, molecules are constantly shaking hands. A protein has to fit into a receptor. An enzyme has to grab a sugar molecule. They have to fit together perfectly to work.
So if a molecule has the wrong hand...
If you have a right-handed molecule trying to shake hands with a left-handed receptor, nothing happens. The connection fails.
Yeah.
It's like they don't even see each other.
I love that analogy. It's like trying to put a left glove on your right hand. You can force it, maybe, but it doesn't function. You can't pick up a coin with it. It's useless.
Exactly. The fit is everything. And this is a modern discovery. To really appreciate this, we have to go all the way back to 1848. We have to talk about Louis Pasteur.
Pasteur. I usually associate him with milk and, spoiling food.
Milk guy, yes. But long before he was pasteurizing milk to save France from bad cheese, he was a 26-year-old chemist obsessed with wine corks. Specifically, the crystals that formed on wine corks, a substance called tartrate.
It sounds mundane, but given who we're talking about, I assume it wasn't.
It was revolutionary. He looked at these crystals under a microscope, and he noticed something that apparently no one else had ever really seen. They had tiny little facets, flat geometric surfaces, and on some crystals, the facets faced left. On others, they faced right.
So he literally had a pile of left-handed crystals and right-handed crystals all mixed together.
All mixed together. And he did something incredibly tedious, which speaks to his genius and his patience. He took a pair of tweezers, and by hand, he separated them into two piles. One pile of lefties, one pile of righties.
Oh, man. That must have taken forever.
I can't even imagine. But then he did the amazing part. He dissolved each pile in water and shone polarized light through them.
And the light, it reacted differently.
The left pile rotated the light to the left, the right pile rotated the light to the right. That was the moment. That was the first proof that molecules have shapes, that they have handedness. Before that, we thought chemicals were just a random soup. This showed they had structure, geometry.
So if molecules have handedness, let's bring this back to biology. Is Earth a lefty planet or a righty planet?
It's a mix, but a very strict, almost fanatical mix.
Right.
Think of it like a construction site. You have the workers and you have the blueprints.
Okay. Workers and blueprints. I like it.
The workers are proteins. They do all the heavy lifting in your body, carrying oxygen, fighting viruses, building muscle, everything. Proteins are made of building blocks called amino acids.
Right.
On Earth, every single living thing, from you to a mushroom to a bacteria in the Mariana Trench, uses only left-handed amino acids.
Only left. There are no right-handed rebels out there. No exceptions.
None that build functional proteins in living systems. If your body accidentally made a protein with a right-handed amino acid, it wouldn't fold correctly. It would be like a worker showing up with a hammer that's backwards. It'd be a disaster for the cell. It gets thrown out.
Okay, so the workers are strictly lefties. What about the blueprints?
The blueprints are DNA and RNA. They hold all the information. They're made of sugars, specifically ribose and deoxyriboose. And on Earth, life uses only right-handed sugars.
That is so specific. It's weird. Why?
That's the billion-dollar question. But let's use another analogy. It's like you imagine a Lego set.
Okay, I'm with you.
Imagine you buy a massive Lego set, but it comes with a strict rule. You can only use the bricks from the left-handed box to build the walls. If you try to jam a brick from the right-handed box in there, the studs don't line up. The whole tower falls over.
So life basically chose one specific set of bricks billions of years ago.
Pretty much. We call it homunculality, same-handedness. Why it happened is one of the biggest mysteries in science. Maybe it was just chance. A 50-50 coin flip that landed heads and everything that came after had to be heads. Or maybe it was some polarized light in the nebula where the solar system formed that favored one over the other. But the point is, life picked the left-handed amino acid box and the right-handed DNA box, and it threw the other keys away.
Which brings us to the big what if, the core of this deep dive.
Exactly.
What if we went to the dumpster, found those other keys, and opened the other boxes?
That is the premise of mirror life.
I want to be clear on the definition here, just so we aren't confusing anyone. When we say mirror life, we aren't talking about a reflection in a glass mirror. I can't touch my reflection.
Correct. A reflection is just photons bouncing back. It has no mass. Mirror life is building a real physical, tangible organism using the other box of Legos.
So we're talking about building proteins out of right-handed amino acids and DNA out of left-handed sugars.
Everything flipped. Every single chiral molecule reversed.
But would it work? Or would it just collapse? I mean, there must be a reason life chose the ones it did.
That's the beautiful thing about physics and chemistry. They don't care about left or right. A mirror ball bounces just as well as a normal ball. Gravity works the same. Electromagnetism works the same. Chemically, there's no reason why a mirror cell wouldn't function perfectly. It would just be backwards compared to us.
I want to visualize these interactions. Let's do a thought experiment. Let's say we have the technology, which I know we'll get to build a mirror human. Let's call him Mirror Mark.
Okay, Mirror Mark. Let's talk about Mark.
Does Mirror Mark look different? Does he walk backwards? Is his heart on the right side?
His heart would be on the right side, actually. That condition exists in nature. It's called situs inversus. But on the outside, looking at him, he would look exactly the same. He walks forward. He talks normally. If you took a picture of him, you wouldn't know. But inside, it's total incompatibility.
Incompatibility how?
Well, let's say you invite Mirror Mark over for dinner. You serve him a nice, crisp apple.
Okay, a healthy choice. Good for Mark.
That apple is full of sugars. normal Earth sugars, which are
Right-handed.
right-handed. So Miramar takes a bite, he chews it, he swallows it. But his body's made of mirror parts. His enzymes, the little scissors inside his stomach that chop up food, are mirror scissors.
And the apple is normal paper.
Exactly. His mirror scissors physically cannot cut the normal paper. The sugar molecules just bounce off his enzymes. They don't fit the lock. So the food would pass right through him. He could eat the biggest feast in the world, digest absolutely none of it, and he would starve to death with a full stomach.
That is terrifying. It's like being a ghost in a way. You can touch things, but you can't interact with the sustenance of the world.
It's a concept that hard sci-fi loves to play with. You might know the Expanse series, specifically the book Cibola Byrne.
Yes, I remember that. They land on that alien planet Ilis, I think.
Right. And it's lush, green, full of life. But the colonists call it the hungry planet because all the life there has a slightly different chirality. The biology is incompatible. They can eat the local berries, but they get zero nutrition. They just vomit it up because their bodies treat it like plastic or indigestible fiber.
It really highlights how fundamental this handedness is. We take it for granted because everything we've ever eaten in the history of our species has the same handedness as us.
We live in a bubble of compatibility. Mirror life steps outside that bubble.
So Miramark starves. That's a hypothetical, but let's get real for a second. We aren't building Miramark anytime soon, but are scientists actually building anything like this? Is this just theory, or is someone in a lab right now holding a beaker of mirror goo?
We are doing it. It's the frontier of synthetic biology. And the answer is yes, people are doing it. But it's incredibly, incredibly hard because of what we call the chicken and egg problem.
Walk us through that. I mean, we've built a lot of things. Why is this so hard?
Think about a cell. To make a cell work, you need a ribosome.
A ribosome, that's the factory.
It's the 3D printer of the cell. It reads the DNA instructions and prints out the proteins. It is the most complex, important machine in biology. Now, here's the catch. The ribosome itself is made of proteins.
Wait, what? So you need a ribosome to make the ribosome?
Exactly. In nature, the mother cell gives a ribosome to the daughter cell. It's an unbroken chain going back billions of years. But if we want to make a mirror ribosome, we don't have a mirror mother to get it from. We have to build the first one from scratch.
It's like trying to build a hammer, but you need a hammer to forge the metal to make the hammer.
Perfect analogy.
Right.
Or imagine trying to build a 3D printer, but the only way to get the parts for it is to print them on a 3D printer you don't have yet.
That sounds impossible. You have to bootstrap it from nothing.
You have to build the tool that builds the tools. And that is what scientists have been doing, piece by painstaking piece, for the last decade. And recently, they've cracked the code on some major parts.
So where are we at? What's the progress report as of 2026?
We have hit some massive milestones. Let's look at the timeline. Back in 2016, a researcher named Ting Zhu made a huge breakthrough. He created a mirror DNA polymerase.
Polymerase, that's the copier machine.
For
DNA.
It's the enzyme that copies DNA. He built a mirror version, chemically synthesized, stitched together atom by atom in a machine, not grown in a cell. And it worked. It successfully copied mirror DNA.
So we have the copier. That's a huge step.
It was step one. Then in 2022, they created a mirror RNA polymerase. That's the machine that reads the DNA to send instructions to the ribosome.
Okay, so we have the copier and the reader. We're getting close to the printer.
We are getting very close. And that's where the estimates from the 2024 report come in. Researchers now estimate we are maybe 10 to 30 years away from a fully self-replicating mirror cell. A mirror bacteria.
10 to 30 years. That is surprisingly close. That's within our careers. I expected you to say a century.
It is remarkably fast. And to show you why this is so mind-blowing and potentially profitable, I have to tell you about the pond water experiment. This is the one that really solidified the why for me.
Lay it on me.
This was a proof of concept to test the durability of mirror biology. Scientists took a paragraph of text written by Louis Pasteur fittingly, and they encoded it into DNA. Just translated the letters into A, C, T, G.
Okay, so they made a biological text file, like a little USB drive made of DNA.
Exactly. They made two batches. Batch A was normal DNA. Batch B was mirror DNA, made with L sugars. Then they took both batches and threw them into a beaker of dirty, slimy pond water.
Pond water, so just full of bugs and bacteria and all that stuff.
Full of nature's recyclers. Bacteria that eat DNA for breakfast. Enzymes that chop up everything they touch. It's basically a biological incinerator.
And what happened?
They came back the next day. The normal DNA, gone, eaten, destroyed. The bacteria had feasted on it. The text file was deleted.
And the mirror DNA.
They came back a week later, still there. A month later, still there. 1 year later, The mirror DNA was perfectly intact, readable.
Wow. 1 year versus 1 day? That is staggering.
Why? Because the bacteria in the pond had right-handed scissors. They literally could not cut the left-handed paper. To the bacteria, that mirror DNA wasn't food. It was invisible. It was just noise in the water.
That is incredible. And also kind of spooky. It's invisible to nature.
It's the ultimate stealth technology.
So this brings us to the why. Why are we doing this? I mean, spending millions of dollars to make invisible DNA, there has to be a payoff.
Well, the payoff is huge. If you can make it work, the benefits are revolutionary. Let's start with medicine.
Benefit number one, indestructible medicines.
Right. Think about when you take a drug. Let's say insulin or a peptide drug.
Yeah.
You inject it or swallow it. What does your body do?
Tries to break it down, right? Digest it.
Immediately. Your body is full of enzymes looking for things to chop up. That's why you have to take pills every day or inject insulin constantly. Your body destroys the drug before it can finish the job. It's a race against your own metabolism.
But if the drug was a mirror drug,
Your body's enzymes are right-handed scissors. If the drug is left-handed paper, the scissors can't cut it. A mirror drug could circulate in your body for days, maybe weeks. It would be invisible to your body's cleanup crew, but, and here's the trick, it could still be shaped perfectly to block a virus or hit a receptor.
so it does the work, but it doesn't get fired.
Precisely. They call them Spiegelmers. It's German for mirror image drugs. There is already research happening here. It could change everything for chronic diseases. You take a pill once a month instead of 3 times a day.
That's huge for patient quality of life. Okay, what else? You mentioned the pond water experiment DNA that lasts a year in dirty water. That sounds like storage.
Benefit number 2, the ultimate data vault. We are generating so much digital data right now. Zettabytes. Hard drives fail. Tape degrades. Servers need power. But DNA is incredibly dense storage.
Right, I've heard of DNA storage. You can fit the whole internet in a shoebox or something.
Something like that. But normal DNA is fragile. If you leave it out, bacteria eat it, UV light breaks it. But mirror DNA, as we saw in the pond, nature can't touch it. You could encode the sum of human knowledge into mirror DNA,
Yeah.
bury it in the dirt, and come back in 1,000 years, and the bacteria wouldn't have scratched it.
That is some time capsule level stuff. We could leave a record that actually survives us. Okay, benefit number 3.
Phage-proof factories. This one is big for industry.
Explain phage-proof. I have no idea what that means.
We use bacteria as factories. We ferment them to make cheese, yogurt, wine, but also industrial chemicals, insulin, biofuels, plastics, giant vats of bacteria doing work for us.
Okay, I'm with you. Little biological workers.
The biggest enemy of these factories is viruses, specifically phages. These are viruses that target bacteria. If a phage gets into your insulin factory, it infects the bacteria and kills the whole batch. It costs billions in lost production.
It's like a flu sweeping through a factory floor. Everyone gets sick and nothing gets done.
Exactly. Now, imagine your factory workers are mirror bacteria.
The viruses, they can't infect them. The keys don't fit the locks.
The viruses act like keys, but the mirror bacteria have backwards locks. The virus bounces off. A mirror factory would be immune to every known virus on Earth. Total immunity.
That sounds like a dream for a manufacturer. Indestructible workers.
It is. It promises efficiency, stability, profit. But
Ah, there's the but. I knew it was coming. The other shoe had to drop.
there is always a but when we talk about rewriting the laws of nature.
Here's where it gets really interesting. Because everything you just said, indestructible, invisible, immune, that sounds great when it's inside a tank or a pill. But what happens if it gets out?
And that is the question that keeps scientists up at night. This brings us to the danger. The invisible invader.
We talked about invasive species before on this show, like the rabbit in Australia.
Right. Introduced for hunting, had no natural predators, multiplied like crazy, ate everything. A total ecological disaster.
Now imagine a rabbit that is bulletproof, poison-proof, and invisible to wolves.
That's a mirror bacteria,
Hmm.
essentially. The catch with mirror life is the exact same thing as the benefit. The fact that nature can't interact with it means nature can't stop it.
Let's talk about the immune system specifically. You said earlier that our immune system relies on shape recognition.
Right. Your antibodies are like right-handed gloves. They feel the shape of a germ. If they feel a spike protein, they grab it and signal the alarm. But a mirror bacteria, its surface proteins are backwards. Your antibodies literally cannot grab them. They would slip right off.
So you could have a mirror infection and your body wouldn't even know it's there.
Your body might not know to fight it. And even if it did try, its weapons, the enzymes it shoots at bacteria to dissolve them, wouldn't work. The mirror bacteria would be wearing armor that our biological weapons can't dent.
That is terrifying. An untreatable infection. What about antibiotics?
Useless. Antibiotics. Penicillin. They work on chirality, too. They attack specific left-handed proteins in bacteria to stop them from building cell walls. They would be completely useless against a mirror strain.
Okay, but hold on. Let's go back to the starvation argument. We said Mirror Mark would starve because he can't eat our food. Wouldn't a mirror bacteria just starve to death if it escaped the lab? It can't eat our sugars or our amino acids.
That is the most common counterargument. It'll just die. And for many things, that's true. A mere lion would starve. But bacteria are resourceful.
But what can they eat that we can't?
A chiral food.
A chiral, you mean not handed?
Exactly. Molecules that don't have a handedness. They're symmetrical. Simple molecules. Glycerol, citrate, minerals. Or, the big one, sunlight.
Photosynthesis. Oh, no.
If we built a mirror cyanobacteria, a type of algae that runs on sunlight and CO2. Sunlight doesn't have handedness. CO2 doesn't have handedness.
So it could grow. It could have all the energy it needs.
It could grow. And because nothing can eat it, no bugs, no other bacteria, no fish, it would have no check on its population. It could just bloom and bloom and bloom.
The gray goo scenario.
The biological gray goo creates what the reports call ecological disruption. It uses up all the nitrogen, all the phosphorus resources that normal life needs. But it doesn't give anything back to the food web. It's a biological dead end. It just hoards resources until the ecosystem collapses.
Okay, that went from cool medical breakthrough to ecosystem collapse really, really fast.
It does. And that's why the scientific community has hit the brakes hard.
So let's talk about the verdict. We're in 2026. What's the consensus? Because surely someone is regulating this. You can't just cook this up in your garage, right?
They are now. It's been a very busy year for bioethics. It started really heating up in December 2024. There was a huge report published in Science. It wasn't just a blog post. It was authored by 38 top scientists, including Nobel Prize winners.
The heavy hitters.
The heavy hitters. And the headline was basically, stop.
Just stop. That's blood.
They drew a very hard line. They said, look, we are making progress too fast. We need to pause. Then, in June 2025, just about 6 months ago, there was a major conference at the Institut Pasteur in Paris.
Paris seems like the right place, considering Pasteur started this whole thing with his tweezers.
Exactly, a full circle moment. And the consensus that came out of that meeting was fascinating. They differentiated between two things, mirror molecules and mirror organisms.
What's the difference? I mean, they sound the same.
Mirror molecules are things like the drugs we talked about, a peptide in a test tube, a snippet of DNA for storage. The consensus is this is allowed, it's safe. A molecule in a tube can't reproduce. If you smell it, it just sits there.
Okay, safe enough. But mirror organisms.
That's a self-replicating cell.
Yeah.
A bacteria, the consensus on that was banned, or at least strongly discouraged and defunded.
Why the hard line? Why not just be careful? Use, like, a super secure lab.
Because of the genie in the bottle problem. Once a self-replicating organism is out, you cannot recall it. You can recall a bad car. You can recall a dangerous toy. You cannot recall a bacteria that reproduces every 20 minutes and is invisible to predators.
It evolves.
It evolves. It spreads. And we have zero antibiotics that work on it. Scientists compared it to playing Russian roulette with the ecosystem. Maybe the gun is empty. Maybe the mirror bacteria are weak and they just die off in the wild.
But if there's a bullet.
If there's a bullet, if they find a niche, if they thrive, the consequences are irreversible. You can't undo an invasive species that you can't see or kill.
It's a sobering thought. We have this technology that could solve so many problems. Storage, medicine, manufacturing. But the risk is essentially infinite.
It is. And that's why right now, in 2026, the door to mirror life is locked. We have the key. We know how to build the ribosome. We know how to build the polymerase. But we have decided, as a species, not to turn that key yet.
Yet being the operative word.
Always.
So what does this all mean? We are standing on the edge of a new biology. We can build a mirror world, but we've decided to keep it theoretical.
For now. But I want to leave you with one final thought. This isn't just about us building life. It's about finding it.
What do you mean, like in the universe?
Think about Mars. Think about Europa, the moon of Jupiter. We send rovers there to look for life.
Right? We look for amino acids. We look for sugars, signatures of biology.
But our sensors.
Yeah.
Our sensors are built to detect Earth life. They are biased. They are looking for left-handed amino acids.
Oh, wow. I see where you're going.
What if we landed on Mars and life there is mirror life? What if Mars chose the other box of Legos billions of years ago?
We would look right at it.
And we wouldn't see it. Our chemical sniffers would report no life found, even if we were standing in a field of mirror bacteria.
We might be blind to half the biology in the universe just because we're only looking for our own reflection.
Exactly. It makes you wonder what else we're missing just because we assume the universe works the way we do.
That is a wild thought to end on. We are looking for mirrors, but only the ones that match us. Well, this was a mind bender.
Always a pleasure.
Thanks for coming along on this deep dive with us. Remember to follow us on your favorite podcast platform, and see you on the next dive.